Okay, well, thank you everybody for being here, and thank you to the organizing committee. My name is Laura Hernández, and I'm going to talk to you about a part of my PhD project, which is entitled Differential Transcriptomic Profiles of Dendritic Cells During Tolerance Induction in Women with Systemic Lupus Erythematosus. So firstly, I'm going to talk to you a little bit about what is lupus. Lupus is a chronic, multifactorial, and heterogeneous autoimmune disease. In this disease is characterized by immune system dysfunction. Among the different mechanisms that are involved in lupus psychopathology, we can observe deficiencies in apoptosis clearance, in the complement system, in the formation of immune complexes, the self-antigen recognition, and all this promotes a loss of tolerance. Tolerance is the capability that our immune system has to detect, to differ what it's mine and what it's not, and to stop self-attack to self-antigens. So this is a key part of the lupus mechanism. And in this context, dendritic cells play an important role. Dendritic cells are the main antigen-presenting cells. They have the ability to take antigens from the outside of the cell and present them to another immune cells. This can polarize the immune response depending on what they sense. And in a normal context, when a dendritic cells finds a self-antigen, this immune tolerance response evits to the development of a self-reactive immune response. But in cases as lupus, this capacity to detect self-antigens is lost. So this leads to overstimulation of immune cells, immune response towards self-antigens, and it can lead to autoimmune processes. In this sense, it's important to acknowledge that there are different subtypes of dendritic cells. For this work, we are particularly interested in a subtype that it's called tolerogenic dendritic cells. These tolerogenic dendritic cells, or TOL disease, have a phenotype that is characterized by an anti-inflammatory profile. They can potentially inhibit active immune responses, which is important for immune activity and autoimmune disease as lupus, and it has made them an important target of interest for the development of immunotherapies. So lupus is also a multifactorial disease. There are several risk factors that have been associated with the risk of having lupus. We can find lifestyle factors, environmental factors, and genetic factors. We have observed that this disease is very heterogeneous. It says that every lupus is different. So it has been reported that there is a higher prevalence, mortality, and disease severity in people with Asian, African, and Hispanic ancestries, such as us. In this map, we can observe, for example, Mexico, that's my country. It has a higher prevalence per year, estimated in contrast with other countries. In this sense, genetic factors have been shown to have an important role in the development of lupus. To date, it has been identified more than 300 loci associated with lupus risk. And they have also identified genes associated with pathways important for lupus physiopathology, such as type 1 interferon, immune tolerance, DNH damage repair, and inflammatory processes. So the question that we are looking for answering is how the genetic factors contribute to this tolerance induction in dendritic cells from Mexican women with lupus. For this, we are working with within the lupus Mexican registry that Ale talked about that in Tuesday. So we collect monocytes from patients with lupus, 22 patients, and 10 healthy controls. We also identify some clinical characteristics such as disease activity, the treatment, the age, etc. From the monocytes, we differentiate the cells into two different phenotypes. The first one, that it's in the upper part of the presentation, it's called monocyte-derived dendritic cells, or modices. These modices are characterized by having a phenotype more inclined to the pro-inflammatory aspect of the dendritic cells. In the other hand, we also stimulate cells to differentiate into tolerogenic dendritic cells that, as I told you before, have this potential to inhibit immune responses. So we stimulate these cells through 8 days, and we recover RNA from monocytes, modices, and toll disease. After this, we did RNA-seq, and we also, well, with this data, we did some differential gene expression analysis. That it's the work that I'm going to present you today. So we have two main biological questions. The first one is, what difference are observed between lupus and healthy controls in each of the cell types that we are recovering? And the other one is, what are the differences observed through the tolerance induction process? So you have seen this heat map. Initially, we didn't see any pattern of differentiation between lupus and controls. So, well, the next question was, did we did well the stimulation? So for this, to acknowledge this part of the process, we select a set of genes previously associated with the three cell types that we were studying: monocytes, modices, and toll disease. And we look for these genes through our data. So we can observe in this heat map that when we look for this set of genes, we can see different patterns across the three different stimulations, or the three different cell types that we were recovering. So the next question that we want to approach was, which genes allow to distinguish lupus from controls? So we explore which cell type specific genes were differentially expressed between lupus and controls. In this part, I present you some of the monocytes differentially gene expressed between lupus and controls. We can observe, for example, the upregulation of FCGR3B, who has been previously associated with the immune complexes clearance that I told you is important for lupus physiopathology. And it has been observed overexpressed in some inflammatory conditions such as severe COVID. In the case of modices, we also observe some genes overexpressed. I highlight FAP-P4 that has been associated with lupus lipid accumulation that can contribute to dendritic cell dysfunction, which is important in lupus, and the reduced capacity to stimulate some T cells. We observed difference in these previously gene associated with these specific cell types, and the cell type that was of particular interest for us is the toll disease. So in this heat map, we can observe that the majority of genes differentially expressed between lupus and controls are downregulated. So what we did to explore which what these genes were contributing to lupus, we look for a previously proposed toll disease signature genes that was observed through a transcriptomic analysis of different protocols to produce toll disease. In this previous work, they established, or they proposed, a toll disease signature of genes. And we observed that it's the third column in the table, that this expression previously observed in toll disease is the same that we are recovering from our controls. But when we look for these genes in lupus, we can observe the opposite effect in the expression of these genes. So we see different in this set of genes previously associated with this phenotype. So we're currently exploring which is the biological effect of these differences. And for this, we are performing some functional enrichment and regulatory network analysis. So the other question that we have is, what are the differences through the differentiation process and the tolerance induction in the case of the toll disease that leads to these differences between patients and control? So for that, we did first differential gene expression, taking monocyte as reference for both modices and toll disease, and this is the result that we have for these comparisons. I'm going to deep in a little bit more. So we explore which genes were differentially expressed between controls and lupus, and this is the first heat map that I'm showing you. And we observed 443 differentially gene expressed between lupus and controls. And when we look for the genes that are differentially expressed between the two differentiation processes, we recovered 944 genes. So in this case, through the differentiation process, we can observe the overexpression in monocytes of IRF4 that is required to this differentiation towards MO disease phenotype. And in the case of Toll disease, we can see some of the markers associated with interleukin 10, sorry, that it's the stimuli we used. And we also can observe the FCGR2A that has been associated, its dysregulation with lupus nephritis, that it's one of the main characteristics and manifestations of lupus. And yeah, we can see differences. So the next step that we're taking is to identify which genes are differentially expressed not only between lupus and control, but also during this differentiation process that can allow us to identify what happens when we induce this Toll disease in lupus, that it's obviously not having an effect as the same that it in the controls. So where are we now? We know that immune tolerance is a key mechanism for the inhibition of autoimmune responses. By exploring this tolerance induction in dendritic cells from women with systemic lupus, we have observed changes in the expression of Toll disease signature genes in lupus when compared to healthy controls. We also identified 944 differentially gene expressed during MO disease and Toll disease differentiation process, and 443 differentially expressed genes between lupus and controls. We identified differentially expressed genes that contribute to important mechanisms involved in lupus physiopathology, such as immune complex clearance, apoptosis, interferon pathways, and anti-inflammatory response. So this work represents a first approach to explore how genetic factors can contribute to lupus tolerance induction in dendritic cells. So far, we have observed difference at expression level between lupus and control in both the tolerogenic dendritic cells and through the differentiation process. And further analyses are needed to depend into the specific genes and regulatory mechanisms involved in this difference, and to explore their biological impact in the lupus physiopathology. So we are currently working in, we're currently exploring which genes allow to differentiate between cell types from lupus and control, which genes contribute to changes in this Toll disease signature expression, and what is their biological effect. And we are also exploring which is the effect of subclinical important feature, such as the treatment and the disease activity. And we're also exploring which are the regulatory mechanisms involved in tolerance induction in lupus. Specifically, we're working on an allele-specific expression analysis during this tolerance induction. To almost finish, last week I heard this phrase that it takes a village, so I want to acknowledge my village. This work led by Dr. Alejandra Medina, the main scientific board of the project, also all the community of patients with lupus that had contributed to this, obviously the medical part of the project, the co-authors of this analysis, also all the students that are the ones to get the samples and are important to acknowledge, and the other part of the team. And well, it's a huge family behind this effort. And finally, I would like to invite you to follow us in social media to know everything that we're doing on the Lupus project. Also, I put some QR codes so you can check out all the work that we are doing. And that's my contact if you have any question. And thank you so much for your attention.
I wanted to ask you if you did some in the pre-processing of the data, if you did some corrections like normally stand for batch or something like that, because you were telling us that the individuals are very different.
Yeah.
This is very different.
Yeah, we did some batch effect correction. I didn't mention, but yeah, we look for that. Firstly, when we did a preparation of the data, and what we have seen, and I'm not presenting right now, it's that these differences between the participants, such as the use of medicaments and the disease activity, contribute to the differential expression. So there are some works that we are currently working on. They are like in pre-analysis, so I didn't want to put it here. But currently with Eve, we are exploring how these variants can be differentiating our populations, because when we use them, we can see some clusters. So we think it's important. Well, thank you so much.